I'm Dr Tracey Martin, Senior Lecturer at Cardiff University School of Medicine. The department I work in is an international collaborative between Cardiff University and various universities in China, and as a whole we're part of the Division of Cancer and Genetics. My research is mainly involved in cancer metastasis, so I'm interested in how cancer is able to escape the primary tumour and move around the body, and particularly with a focus on how it's able to pass the blood-brain barrier and form secondary tumours in the brain. So our current project is using the Lumit Immunoassay cellular systems to assess the expression of a motility protein called NWASP that we think is important during the metastatic cascade. It's a Goldilocks molecule. It can't be too high or too low. The expression has to be just right, and that's what we were interested in assessing with the 150 novel compounds that we've discovered. So we're actually going through a sort of drug discovery process to find the best 5. Originally, we were just using Western blotting to look at the levels of the protein to see if it was too high, too low, whether it was phosphorylated or not. But this was extremely labour intensive. It took a lot of time. It's an expensive process. We wanted something that had more high throughput, that was more reliable, consistent, and that we could do lots of replicates with. And the 96-well format of the Lumit is perfect for this. So the Lumit assay provides advantages in high throughput, consistency. As I said, we have 150 compounds that we've been investigating. If we're looking at 150, lots of different concentrations and different cell types, that's incredibly labour intensive, and the Lumit system offers a faster, more reproducible technique for looking at these. So far, this work has shown that a number of the compounds we have are effective at regulating the expression of NWASP, and that we can now narrow them down to 5 potential therapeutics that we can take forward for further research. Next steps of the project then are to focus on the 5 of the 150 compounds that we have for further investigation. We've looked at the expression. We now need to look at how these are able to control cellular processes of breast cancer cells, and whether it's effective against the propensity for migration and metastasis. In the future, we hope that this research would translate into a new breast cancer therapy. We are desperately looking for therapeutics for cancer, particularly breast cancer, and hopefully that we'll be able to take this into the wider cancer market, particularly as anti-metastasis therapeutic and in other cancer types, and ultimately my particular interest using it as a therapeutic against cancers that have already spread across the blood-brain barrier.